Good morning. Myself, Dr. Balasubramaniam, working as Assistant Professor, Department of Zoology, Government First Grade College and PG Center, Chintamani. Today, we are going to discuss about the cellular organization in the case of the sycon. Now, cellular organization in the case of sycon, it belongs to the first semester BSc syllabus of the Bangalore North University. Learning objective of the topic: to understand the cellular details or histology of the sycon. To know the cellular organization of the sycon. Session outcome: the students will be able to understand the cellular details of sycon and also know the histological details of the sycon. Now, previous classes we discussed about the morphology of sycon and externals of sycon. Before these classes, we also discussed the general characters of sycon or the porifera. ಈಗ cellular details of sycon. Now, sycon is a organism which shows the outer pinacoderm, and this pinacoderm consists of pinacocytes. The pinacoderm consists of pinacocytes, and inner coenoderm, which consists of inner coenocytes. So, in between the outer pinacoderm and inner coenoderm, we have the mesenchyme. This mesenchyme is a non-cellular, non-gelatinous layer, and in the mesenchyme we find different type of cells. ಈಗ outer pinacoderm ಅಲ್ಲಿ there is a pinacocytes. Inner coenoderm ಅಲ್ಲಿ we have the coenocytes. So outer pinacoderm and coenocytes, between these two layers we find the mesenchyme, and the mesenchyme consists of the following types of cells, namely archosites or amoebocytes, colonocytes, chromocytes, thysocytes, myocytes, scleroblasts, gland cells and germ cells. ಈಗ ಒಂದೊಂದಾಗಿ ನೋಡೋಣ. ಈಗ pinacocytes are spindle shaped cells and are found in the epithelium of sponges. They are found in the epithelium of the sycon. They play a very important role in contracting and stretching, cell adhesion, cell signaling, and phagocytosis, and also polarity and the direction. Pinacocytes are filled with mesophyll, which is a gel-like substance which maintains the shape of the structure of the sponges. ಈಗ pinacocytes are the outermost covering of the sponges. So they help in covering of the sponges and help give protection. So that is the main function of the pinacocytes, is to give protection. ಈಗ second layer, that is the coenoderm, which has the coenocytes, and coenocytes are round or oval cells which have the round or oval body. They possess nucleus and one or many vacuoles in its cytoplasm. The free end of the cell body has a comparatively long flagellum, and the base of the flagellum is surrounded by contractile, transparent, collar-like outgrowth of the cytoplasm. ಈಗ coenocytes main function to help in the nutrition. The coenocytes are lining the spongocoel. Spongocoel ಗೆ layering coenocytes ಇದೆ. Now these coenocytes are also called collar cells, and they help in the nutrition. Now we know that poriferans, the digestion is intracellular. So digestion takes place inside the cell. So that is in the coenocytes the digestion occur. Next, archosites. See, these archosites is the ಮೂರನೇ type of the cells histologically. Archosites, so these are the undifferentiated embryonic aminocytes which are large in size and the nucleus show distinct nuclei. So archosites are also called stem cells. These archosites are also called totipotent cells. Now these archosites in the ಬೇರೆ ಸೆಲ್ಸ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ. ಯಾವುದೋ ಸೆಲ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ, ಪಿನಾಕೋಸೈಟ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ, ಕೊಯಿನೋಸೈಟ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ, ಮತ್ತೆ ಆರ್ಕೋಸೈಟ್ಸ್ ಇಟ್ ಸೆಲ್ಫ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ, ಮತ್ತೆ ಕ್ರೋಮೋಸೈಟ್ಸು, ಮತ್ತೆ ಥಿಸೋಸೈಟ್ಸು, ಜರ್ಮ್ ಸೆಲ್ಸು, ಗ್ಲ್ಯಾಂಡ್ ಸೆಲ್ಸು ಇವೆಲ್ಲನೂ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ. ಎಲ್ಲಿಂದ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ? ಆರ್ಕೋಸೈಟ್ಸ್. ಸೊ ದ ಆರ್ಕೋಸೈಟ್ಸ್ ಆರ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಅಮೀಬೋಸೈಟ್ಸ್ and these are called totipotent cells. See, totipotent cells andre they can differentiate to any other type of the cells. So that is the meaning of the totipotent cells. They can differentiate to any other type of cells. So that is the importance of archosites. Archosites, nodi, archosites they are undifferentiated embryonic amoebocytes which are large in size and their nuclei show distinct nuclei. They are totipotent in nature and they can transform to any kind of cells needed by the organism. They play a role in digestion, eliminating the waste, and also can give rise to the sperms and the ovum. So that is the very important characteristics of the archosites. ಈಗ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ಡಿಫರೆಂಟ್ ಕೈಂಡ್ಸ್ ಆಫ್ ದಿ ಸೆಲ್ಸ್ ಇನ್ ದಿ ಸ್ಪಾಂಜಸ್. ಈಗ ಒಂದು ಎ ಇಸ್ ಎ ಪಿನಾಕೋಸೈಟ್ ವಿಚ್ ಇಸ್ ಆಲ್ಮೋಸ್ಟ್ ಸ್ಪಿಂಡಲ್ ಶೇಪ್ ಸೆಲ್ and B is a porocyte which has the nucleus and C is the coloncytes which has the pseudopodia and D is the chromocyte which has pigments and E is the tropocyte and F is the archosites and G is the scleroblasts from which the spicules are generated and I is the gland cell while H is the myocyte. So the myocyte help in contraction and relaxation. Iga other cells, so coloncytes. See, coloncytes, most of the other cells are smaller and stellate shaped and possess radiating process. So these are called coloncytes or connective tissue cells. They remain fixed by the cytoplasmic process. So they help in giving the definite shape to the sycon. Even next to chromocytes. See, these are pigmented amoebocytes with lobose pseudopodia. That means they give definite color to the sponges in general and sycon in particular. Next, thisocytes. These amoebocytes are storage cells with a lobose pseudopodia. So these are storage cells which store some food materials. Myocytes are fusiform cells, highly contractile cells found around the osculum, apophyles and other pores. They may be the dermal ostia. Dermal ostia allino myocytes ide. So they allow the entry or exit of the water. Because of their contractile nature, they allow the entry and exit of the water. These cells are arranged in circular fashion and act as a sphincter to regulate the diameter of the openings. So water flow through the osculum is regulated. Next, to gland cells. These cells are found attached to the body surface by long strands and secrete some mucous substances so that this sycon gets attached itself. Now we know sycon is the sedentary form or sessile form. It needs some material to get attached itself to any other substratum. So it is these gland cells which are going to secrete some substances so that they attach to the substratum. You know the 10th one, no? That is a gonads. So these amoebocytes can also differentiate to the germ cells. Here we call them the germ cells. They can differentiate to the ovum cells or they can differentiate to the sperm mother cells. So from these sperm mother cells, the sperms are arising, and from the ovum mother cells, the ovum are arising, or the eggs are arising. Here ನೋಡಿ ಇಲ್ಲಿ sycon diagram is there. Now we have the coinocytes or collar cells, and porocytes and mesohyl, that is a mesenchyme, spongocele, spicules, pinacocytes and ostium. Now recap of the study. We understood the cell structure of the sycon, and also we know the histology of the sycon. ಈಗ ಮಲ್ಟಿಪಲ್ ಚಾಯ್ಸ್ ಕ್ವಶನ್ ಅಲ್ಲಿ mesenchyme of sycon has a dash which are totipotent in nature. So the archosites or amoebocytes are totipotent in nature. ಈಗ ಪಿನಾಕೋಸೈಟ್ಸು ತಪ್ಪಾಗುತ್ತೆ, ಕ್ರೋಮೋಸೈಟ್ಸು ತಪ್ಪಾಗುತ್ತೆ, ಕೋಲನ್ಸೈಟ್ಸ್ ಆಲ್ಸೋ ಇಸ್ ಎ ರಾಂಗ್ ಆನ್ಸರ್. ನೆಕ್ಸ್ಟ್, ಡ್ಯಾಶ್ ಆರ್ ದಿ ಪಿಗ್ಮೆಂಟೆಡ್ ಸೆಲ್ಸ್. ಸೊ ದೀಸ್ chromocytes are the pigmented cells. So thisocytes or pinacocytes or coloncytes are wrong answer. Flagellated collar cells are also called porocytes, amoebocytes, amoeboid cells. The coinocytes are scleroblasts. Coinocytes are the flagellated cells. So these coinocytes are found in the coinoderm. Dash are the fusiform contractile cells. So myocytes are the fusiform contractile cells. Pinacocytes ತಪ್ಪಾಗುತ್ತೆ, coinocytes ತಪ್ಪಾಗುತ್ತೆ, spicules ತಪ್ಪಾಗುತ್ತೆ. Apparent diploblastic condition occurs in the phylum Porifera. That is the correct answer. So Coelenterata absolute diploblastic condition ಇದೆ. Protozoa diploblastic condition ಇಲ್ಲ. Platyhelminthes ಅಲ್ಲಿ we have diploblastic condition. Okay, ಇನ್ನೊಂದು ಪಾಯಿಂಟ್ ಇಲ್ಲಿ ಮರೆತೋಗಿತ್ತು. ಈಗ ಸ್ಕ್ಲಿರೋಬ್ಲಾಸ್ಟ್ ಆರ್ ದಿ. ಸೊ ಸ್ಕ್ಲಿರೋಬ್ಲಾಸ್ಟ್ ಆರ್ ಅದರ್ ಟೈಪ್ ಆಫ್ ಸೆಲ್ಸ್. ಅದು 11 ಟೈಪ್, 10 ಟೈಪ್ ಆಫ್ ಸೆಲ್ಸ್ ಆಯಿತು. ಸ್ಕ್ಲಿರೋಬ್ಲಾಸ್ಟ್ another type of cells. So this scleroblast are spherical or oval in nature, and these scleroblast give rise to spicules. They give rise to spicules. The spicules in these sponges, particularly ಈಗ ಇದರಲ್ಲಿ sycon ಅಲ್ಲಿ, it may be the monoaxon spicules, triaxon spicules or tetraaxon spicules. So the spicules are arised from the scleroblasts. So this is the 11th type of the cellular details. Okay, I hope you understood the histology of the sycon. Thank you.